Welcome everyone to this Rocha talk, stories from Rocha's conservation work across Canada. My name is Rick Faw and I serve as the VP of Programming for Rocha Canada.
And I am Christy Jouteau. I'm the Conservation Science Director for Rocha Canada. And I just wanted to take a moment before we begin to acknowledge how grateful I am, Rick and I are, to work out of a site that is within the unceded traditional territory of the Coast Salish peoples, including Semiahmoo, Kwantlen, Stolo, and Saanich First Nations, who continue to steward these lands to this day. And I just would invite you to consider and acknowledge the Indigenous traditional territories that each of you are on, wherever you may find yourselves this evening as well.
For those of you that are new to Rocha, the name means the rock in Portuguese. We got this name because the first Rocha project began in Portugal in the early 80s. Nowadays, Rocha is this global network that consists of Christians caring for creation by working through community-based nature conservation projects. And this takes place in about 21 countries around the world, actually 22 now. Sweden just officially joined. And in each place, it's adapted to the needs of the diverse bioregions or watersheds of those particular places. You can learn more about this work at rocha.org. In Canada, Rocha Canada, our vision is to transform people and places by showing God's love for all creation. And we currently have 3 study centers in kind of South Surrey, Greater Vancouver in BC. We have one in Hamilton, Ontario, and one just outside of Winnipeg in Manitoba. As well, we have some smaller partnered projects in other parts of the countries, one of which is you'll hear all about today. And these 3 centers and these other projects, they each demonstrate our commitments in diverse ways, but hold a common set of values.
So to give you a taste of what that looks like on the ground, if you were to visit one of our project sites, you might find some biologists researching sensitive habitats or species at risk, actively working towards restoration of biodiversity and ecological health, or you could meet an education team getting different age groups all the way from preschool to adults outside and actively learning about how nature works in forests, rivers, wetlands, agriculture. You might see a team of people growing vegetables in ways that foster soil health and biodiversity. And you would find staff, volunteers, interns working together to try and live more lightly on the land. And seeking to humbly embody how our faith in a living, loving creator God can shape our lives and our lifestyle.
Let me just say before we move on that if you are new to Rocha Canada, please do check out our website, rocha.ca, and as COVID allows, visit one of our projects. The Zoom media allows us to do all sorts of things, but we really are more native to in-person interaction. So we'd love to have you come visit. In the meantime, please reach out if you'd like to know a little bit more. I put my email in the chat. There we go. And by the way, I also put a little schedule for this evening in the chat. For those of you that have received that schedule multiple times, that's because people continue to arrive. So we're just accommodating the latecomers. So that's a little bit about us. Speaking of the schedule for tonight, what's on tap today? So this is our latest installment of Rocha Talks. This is number 9, I think. So there are a number of others that you can find on our Rocha YouTube channel. It's a series of online events where we have open dialogue about core issues of faith as it relates to our life in this world of creation and of being human creatures. Tonight, we plan to delve deeper into some of the conservation science work that Rocha is doing all across our projects. And Christy, you've been facilitating in-person events that showcase what we're learning about creatures and watersheds and in our backyards for a number of years now. but I'm really excited by this chance to do this online. This is the first time we've done this online because it opens up this possibility for so many more people to join us in this learning. I know Christy, you've got a great lineup for us tonight. So can you tell us a bit more about what to expect over the next 80 minutes or so?
Yes. So our plan for tonight is to give you a taste of some of our conservation science projects across Canada. So we will virtually visit each of our Rocha hubs and explore different levels of biodiversity, highlighting the ways that we're studying and caring for creation in those places. We'll start by focusing on a few vertebrates, so barn swallows, toads, and salmon, all in BC. And then we're going to pause for a few questions from a delegated respondent. So I've asked Andrew Bayless, a past conservation biologist for Rocha in BC here, who's now working for DFO, to ask a few questions of those three presenters. Then we're going to move on to three more presentations from Ontario, Manitoba, and BC, with a few more questions from a different delegated respondent, Rachel Krause, who's one of our science advisory team members based in Manitoba. And then at the end, after those few questions, we're going to open it up to the floor and answer questions from the chat. So anytime you have a question, feel free to just pop it into the chat, and Lainey behind the scenes is going to be compiling those, and we will have time to ask them at the very end. And just to describe how the presentations are going to be working tonight, we're using a Pechacucha style format, which we've used in the past in our in-person presentations. It's 20 slides for each presentation, 20 seconds each, so it moves through them fairly swiftly, and it keeps the presentations punchy with lots of visuals to help facilitate hopefully an engaging sharing of information.
That sounds great. I'm looking forward to this. I have done Pechacucha presentations before, and it is tricky. So thank you to all our presenters for the work it takes to conform to this format. It is a challenge. So well done, and I'm looking forward to it. I think that's all I've got to say. Christy, can you take it away, and we'll begin.
Okay, sounds good. Thanks, Rick. So first, we're going to hear from Michelle Jackson, and she is a conservation research biologist on the BC team based in South Surrey. She's been with us for the past 4 years, and she brings a strong academic background to the team, has been leading our barn swallow research for these past 4 years. I'm just going to share about that today. For it, Michelle.
All right, can everyone see my screen?
Looks good.
Good, okay. Well, good evening. It's great to be here and see you all. So I'm going to be talking to you about Rocha's barn swallow nest monitoring program here in BC, which began in 2014 and continues today. As this program has evolved, we've begun to incorporate more data collected by volunteer landowners or citizen scientists, some of which might even be here today. And partnerships with private landowners play a really important role in this project and in Rocha's work overall. And citizen science is something that we hope to continue to build on in the future. So barn swallows are perhaps the easiest swallow to identify. They have a long forked tail and orange undersides. They're incredible acrobats, and they forage for insects over open fields. They build cup-shaped nests out of mud, and they're reliant on barns and other outbuildings for nesting, along with open space for foraging, both of which are in decline. They exist on every continent except Antarctica, and they migrate from their breeding grounds in the north to overwinter in the south every year. And here you can see their summer range in yellow and their winter range in blue. But despite this global distribution, they have been federally listed as threatened in Canada since 2017. And since the mid-1980s, barn swallows have declined by 80% in Canada and 87% here in BC. And this is due to complex interacting effects of habitat loss, including development of farmland, global insect declines due to pesticide use, climate change, and also competition with some invasive species. And because of these threats, Rocha started monitoring barn swallow nests at 10 sites in the Little Campbell River watershed in 2014. One of these sites included a barn at Brooksdale in the lower left photo. And our goal was to better understand how barn swallows are doing locally and to begin compiling a long-term data set on nest success. So what does nest monitoring look like? Basically, we count the number of eggs and chicks in each nest, and we estimate laying, hatching, and fledging dates. To monitor low nests, we use a mirror attached to a pole, as you can see on the left. And for higher nests, we have a longer pole with a camera attached, as you can see in the photo on the right. And when the chicks first hatch, they're just little pink blobs. They're very hard to count, as you can see in the top right. There's actually one egg in there that's still unhatched. And as the chicks get older, they start poking their heads out, and then we count them from the ground to minimize disturbance. And on the bottom right there, you can see some new fledglings. They're still hanging around the nest site and looking curious. Here you can see the number of buildings with active nests that were surveyed each year, as well as the total number of nests. The barn pictured here was one of our most productive sites with 10 to 20 active nests, but it was unfortunately lost to a fire in 2019, resulting in that drop in nests that you see after 2019. This map here shows nest sites across the watershed. In 2016 and 2019, we mailed 100 letters around to landowners asking whether or not they had barn swallows nesting on their properties. Some of you here tonight may have been a recipient of one of those letters. And this resulted in a few additional sites, both directly and through word of mouth among neighbors. So we've been trying to increase the citizen science component of this project by providing training and equipment to people who are interested and able to monitor their own nests. And this allows for more landowner engagement with the project and with conservation, and it helps us collect a lot more data than we would otherwise. And this year, 8 landowners monitored nests for us, many for the 3rd year in a row. And I think this is a really important aspect of this project because it connects people directly with the birds on their property. And landowners seem to really enjoy watching the swallows. They often will get their families involved, and some will regularly send us pictures and anecdotes about what the swallows are up to. And here you can see different measures of nest success over time. 2017 was a pretty poor year with low clutch survival. It also had the lowest minimum temperatures for May, which was when most first brood chicks were in the nest. So cold snaps might have played a role in those nestling deaths that year. Sadly, we have noticed a substantial decline in nest activity over the 7 years. This barn at Brooksdale, along with several other sites, have had total loss of nesting barn swallows over time. And it's a common story among landowners that sites used to house many swallow nests and now they have few to none at all. So we'd like to better understand factors that influence nest success, both at the nest and the site level. So we measure several variables that might influence nest success, like height of the nest, opening, location within the building, level of human presence, livestock presence, and presence of other nests within the building. And we've also classified land cover within 400 meters around the nest sites, which is the average distance that a swallow would typically travel to forage. This is a fairly coarse map showing 4 land cover categories. This year, we have a master's student working on classifying land cover at finer scales. So that map will be forthcoming. And initial analyses have shown that buildings are more likely to have active nests when they're surrounded by less forest and more freshwater. And less forest typically means more open space for foraging, and freshwater is really important for aquatic insects, which have been found to be really nutritious for swallows. So what's next? We want to continue engaging landowners through citizen science. We also want to build up partnerships with scientists who are actively working to better understand and reverse swallow declines and contribute our data to larger projects. And we'd also like to focus effort on habitat enhancement for swallows locally. For example, we're exploring the use of artificial nesting structures that might attract swallows, such as this structure in Rondeau National Park in Ontario. And we've been in discussion with a biologist who's in the Columbia Valley, who's testing some designs this summer that we might try to replicate here at Brookesdale. Other habitat enhancement measures that we recommend are protecting and restoring ponds and wetlands, planting hedgerows and native gardens to attract insects, and whenever possible, maintaining those old farm buildings and allowing swallow access in and out through doors and windows. So to summarize, we know that this area still contains some high quality habitat for barn swallows, but swallow habitat isn't valued economically, especially as development pressure intensifies. And climate instability and limited knowledge about overwintering habitat make the problem even more complex. But our hope is to better inform landowners and decision makers about the plight of the species while testing out conservation measures that can be implemented locally. So with that, I'll just say thank you to all who have contributed to this project, and thank you all for listening.
Thanks so much, Michelle. And again, if you have any questions, feel free to post them in the chat. And now we're going to hear from Laura Tsai. She is our full-time biologist based out of Brookesdale in South Surrey. And she's also been with us for about 4 years and oversees the intern research and restoration projects to improve the health of the Tataloo or the Little Campbell River watershed. And so over to you, Laura.
Thank you. Well, my name is Laura Tsai. I am one of the conservation biologists out of Rocha, Canada in BC. And this evening, I'm going to share a little bit about our Western Toad project in the Little Campbell River watershed that was started in 2013. And we continue to monitor to this day. We have lots of staff and volunteers and interns that have been helping us. The Western toad is a true toad, meaning it has soft, dry, bumpy skin and is toothless. They are brownish in color and have a cream-colored stripe down their backs. They also have a parotoid gland behind the eye that excretes toxins when the toad is stressed. The Western toad inhabits both terrestrial and aquatic habitats during different life stages. As adults, they spend most of their time foraging and hibernating in mature forest ecosystems. Aquatic ecosystems are also used by adults to breed in and for tadpoles to rear in, and for the juveniles to migrate from. In February, adult toads emerge from hibernation in forest ecosystems and begin to migrate to their natal breeding ponds. Through March and April, they court and spawn in the wetland. July through August, tadpoles metamorphose and emerge from wetlands and begin migrating to terrestrial ecosystems. And in September, toads start to enter into hibernation. Toads become sexually mature between 2 to 6 years of age. Females will only reproduce every 1 to 3 years or once in their lifetime, whereas males can mate every year with multiple females per year. One individual female can lay up to 12,000 eggs in a string-like formation in shallow, warm water during the breeding season. After 3 months of rearing in the ponds, the juveniles grow limbs and can emerge out of the water. They immediately start their migration to mature forest ecosystems. This migration draws a lot of attention as they migrate en masse, and thousands of toadlets can be seen moving together across the land. Our Western toads in BC are being stressed by several factors. One key stressor is increases in urbanization and an increase in roadways being built through the middle of their habitats. Oftentimes, toads have to cross one, if not more than one large roadway in order to access breeding ponds and terrestrial forests, putting them at risk of being run over. Other stressors affecting toads include competition for resources by introduced fish and amphibian species, as well as increased predation by introduced species, the introduction of diseases such as chytrid fungus, and the use of pesticides and fertilizers in the surrounding ecosystems. So why do we care so much about these toads? What are their role in the ecosystem? First, they can be used as a natural pest control, as they are often found in gardens feasting on unwanted insects. They are a source of food for other wildlife species that can handle their toxic skin. And third, they aid in the dispersal of nutrients between aquatic and terrestrial ecosystems. We have been conducting annual monitoring surveys on population of Western toad from 2 connected wetlands since 2013 in the watershed. We have looked at the timing of emergence as well as the direction of migration. We have observed that this population moves in a northeast direction and disperses into the forest habitat, which can be seen by these arrows. In 2018, we started using transects and quadrats to monitor the number of toadlets that are being observed crossing the roads surrounding the breeding wetlands. We were able to identify high mortality hotspots due to car traffic on several roadways. This is outlined in red and orange on the map. Most mortality was observed on 200th Street. Due to these findings, we decided to build a fence to direct the toads through a culvert under the road on 20th Avenue and keep them away from 200th Street. This map shows that the fence is effective, yet not perfect, as many toads are still able to get around the fence and cross the road on 20th Avenue, increasing mortality. Here's another figure that shows the data. The figure on the left is from 2020 when no toad fence was built. The yellow and red areas show where toads are observed crossing roads. The figure on the right is from 2019 when a fence was built and shows that the toads are being prevented from crossing 200th Street and therefore decreases overall mortality on the roads. This table is a summary of the number of toads observed, both dead and alive, along the road surface during migration surveys from 2019 to 2021. We can see that the number of dead toadlets is greater in non-fence years compared to fence years. Our 2021 data is still being analyzed as the toads just finished their migration this year. We have been working with the Township of Langley on this project as well. They have added toad crossing signage and post local access only signage during toadlet migration to help decrease the amount of road mortality due to car traffic. We have yet to determine whether the signage is succeeding in reducing car traffic, but we hope it makes people aware of the migration. In 2018, we also started to monitor the adult migration to the breeding wetlands in early spring. We would go out on rainy evenings after sunset and we would record the GPS location of observed toads along the same roadways as the juvenile surveys. The main goal was to determine if there were any main corridors that the adults were using to travel through. For the most part, the observed toads were scattered along 4 of the 5 road transects. However, a few corridors can be highlighted in the more forested patches. We've also been looking into how to generate a population estimate. This can be done by comparing the number of observed toads to the number of recaptured toads. Every time we observe a toad, we take a picture of the unique markings on its back. These pictures are labeled and stored. The pictures can then be compared with one another to determine if the same toad has been observed multiple times. Since 2013, we have only found 1 breeding population of western toad in the watershed. However, this summer we heard about a second population of toads in the watershed. This population is just northeast of the current population that we've been monitoring. It's not a new population. It has been in the area for many years, but we hope to study it in future and help protect them as well. I want to say thank you to the township for their help and to all of our volunteers, to Penny, who graciously lets us build the fence on her property, and to all of the interns for their hard work over the years. Thank you.
Thanks, Laura. That was awesome. And I'm just gonna keep us moving along here. So next we're gonna hear from Cindy Verbeek. She is based in Houston, BC, and has been champion Rocha work in the North for longer than any of us. And she's been building a team around her and has inspired countless folks to care for creation in the Upper Bulkley River watershed. I'm excited for you all to hear about the amazing things that she's been doing. Take it away, Cindy.
Thank you, Christy. Just share my screen here. There we go. So yeah, good evening. So my name is Cindy Verbeek, and I have lived and worked for, lived in Houston and worked for Rocha for the last 18 years as the Northern BC project coordinator for Rocha Canada. Today I'll be sharing about our work with salmon as stream keepers and our hopes for science into the future. I have to admit, I am an educator, not a scientist, so we'll give it a try. Houston, we are not in Texas. We are in Northern BC. We're halfway between Prince George and Prince Rupert. We are a logging, ranching, mine, fishing, and hunting town of about 2,500 people, and the closest city is about 3.5 hours away. And we live in the Upper Bulkley River watershed. I am privileged to run a small coho hatchery and nature center, and have been loosely doing conservation science and citizen science work in the Upper Bulkley for the past 7 years. And have also been raising coho salmon in our hatchery. Our hatchery is what we call a stewardship hatchery, which has the main goal of education and community engagement. We raise up to 10,000 salmon each year. But to raise fish, we need to know about the rivers that we will be returning them into, which is where the stream keepers work comes in. Excuse me. About 7 years ago, we invited Zoanne Morton from the Pacific Stream Keepers Federation to come train us in stream keepers methodology, including habitat assessment, water quality monitoring, invertebrate surveys, spawner surveys, and riparian planting, and of course, public engagement. One of my favorite things is the invertebrate surveys. I love insects, and so over the years, that has been the main focus of the research that I've done, and we have thankfully always found lots of species diversity of these big three, the caddisfly, stonefly, and mayfly, that are key indicator species of a healthy stream because they're the first to succumb to pollution. A lot of the science work is done in collaboration. And this is a cabin course that I was able to take that was sponsored by the Office of Wet'suwet'en, on whose territory we live and work. And it's been great to work with them. Some of the limiting factors for salmon in our river, in fact, the limiting factor is the temperature. Over anything above 15 degrees Celsius, salmon health becomes a concern, and our monitoring has shown that our river can get up to 24 degrees in the heat of the summer, which is a huge issue for both juveniles and adult salmon. This is Bob from the Department of Fisheries and Oceans checking one of the 14 temperature loggers that I helped the Department of Fisheries and Oceans find good places for and install, and they have shown, as we suspected, that rising water temperatures are a huge issue in our watershed. The other big issue is oxygen levels. As the water warms, it loses its capability of keeping oxygen. In this case, this was a stream that would normally be flowing, but because of a very hot summer and poor management upstream, the pool was starting to dry up, leaving over 1,000 coho, chinook, and steelhead fry stranded. Because we had been monitoring the stream, we were able to mobilize volunteers to get these fish back into the main stem of the river, where they could find deeper, cooler pools and survive. Another issue is beavers, and in this pool here, this pool was drying up because the beavers had built a dam and the water was being diverted away from the main part of the river. And if we hadn't been out monitoring the river, we would never have found this group of fish and never had the opportunity to put them into cooler water. Beavers are a good thing, and yet they can cause problems as well. Each year we walk many kilometers of river doing spawner surveys beginning in mid-August and going until the mid-October. One of the advantages of this is that this river is not allowed to be fished on, and so we are really sometimes the only eyes on the river to see what's going on. So not only are we looking for our brood stock for the next year, like this beautiful salmon, but we're also looking for obstructions to spawner migration, issues along the riverbank that we might be able to do something about. Like I said, beaver dams is one of those issues. And if the spawners are stacked up behind it, we will go in and breach a small section of it to create a way through. This isn't, of course, a permanent solution because the beavers will be in there the next evening and fixing it up, so we have to go back the next day, but at least those fish can get through. And we have compromised in this way. Culverts cause a huge issue for salmon migration, like this hanging culvert. there's no way the fish will get up there. And I've been working over the last couple of years with the province of BC to help identify culverts in our watershed that need restoration or need changing. And then the scientists can go in and do some studies and make recommendations. And of course, we're always looking for human interactions with the river. Quadding is a big sport in Houston, and it's so much fun to drive through shallow water or sometimes even flat, dry riverbeds. Unfortunately, it also means that you're driving over salmon redds, because this is exactly where the salmon would be spawning. But there's lots of opportunities for restoration and for more research. I've had the privilege to work with the Wet'suwet'en First Nations out of Burns Lake on bank stabilization projects that has made the difference between losing 50 feet of riverbank in a farmer's field 1 year to 5 centimeters the year after the stabilization work. So this stabilization is not only helps keep sediments out of the streams and protects the salmon's gills, but it also captures sediments and creates salmon spawning and rearing habitat. So it's really a win-win-win situation all around. I'm hoping to be able to do more of this kind of work in the future. We are looking at possibly hiring someone who has more scientific background than I do. And so possibly the next presentation will be much more robust. Thank you for your time, and the salmon, thank you for your support.
Thanks, Cindy. That was fabulous. And very robust of a presentation, I must say, for a non-scientist, which you keep calling yourself. You do such good work. Excellent. Thank you all three. I'd like to invite Andrew Bayless now to join us to ask a few questions of our first three presenters. So we could have all three presenters just put their video on, and yes, I'll leave it to you, Andrew. Thank you.
Thanks, Christy. Thank you everyone for your presentations. You all did very, very well. It's super encouraging to see all of this work being done. So I have a few questions, one for each of you. So I'm just going to go in chronological order. So I'll start with Michelle. Thanks so much for your presentation. It's really encouraging to see how the Barn Swallow project has evolved since 2014, so 7 years, I guess, and 4 years with you at the helm. So I just really appreciate your work with citizen science and looking at the overlap with habitat restoration and how that might benefit the swallows. My question for you is, what is one of the most interesting things that you've observed or learned over the 4 years that you've been working on the project? So I'm thinking things like unexpected behaviors that you've seen swallows do, or unexpected factors that have affected nest success, or changes in the landscape or interaction with predators, that sort of thing. And that's kind of the vibe I'm thinking about. Curious what you have to offer about that.
Sure. That's a hard question because I don't know, I'm constantly learning new things and it's hard to pick one. Just the thing that pops into my head right now, because I was just at a conference hearing a lot about this, is just the importance of aquatic insects for swallows. I mean, it kind of makes sense, but I just never realized how much more nutritional they are for swallows in particular than terrestrial insects, which really points to the importance of preserving wetlands and ponds on the landscape, which we know is important for many reasons, but being able to really make that connection with the swallows, I think is really interesting and also really, just really valuable information to have as conservationists. It gives us one more thing to point to say, you know, we need to protect these wetlands because not only is it protecting amphibians and fish and other things, but it's also protecting swallows. And there's just so many linkages between swallows and so many other things, like with insect declines. And yeah, just, I don't know, that's the thing that kind of first pops into my head. So yeah, I guess I'll
great.
leave it at that.
Yeah, thank you, Michelle. It's really cool to learn about those interacting ecosystem features, and especially just thinking of all the habitat restoration work that Rocha does and how that might overlap with properties that are monitoring barn swallows as well. So thanks again. So Laura, moving on to Western toad. Thanks for your presentation as well. I was really interested in those hotspot maps that you put up, showing the effectiveness of the fences and how they sort of diverted the toads away from 200th Street, or at least that's what it appeared in 2019, I believe. So that's, I feel like that could be really well applied at a more broad scale. And I think some people have sort of brought that up in the chat as well. And also the photo identification technique that you mentioned is super interesting. And I wanted to ask about that. So I'm curious if you've actually had some success in matching individuals from one survey to the next, or between different surveys. In my head went like, could we create like a Shazam for toads? You just take a photo and it tells you who it is. But I know someone also mentioned some software in the chat as well. So I'm just curious if you have any thoughts about photo ID for toads.
Yeah, so in the spring during our adult surveys, we found 29 toads. So that was actually quite high for us. And there is some software that is being developed. There's a group in the interior that's using, I believe it's MATLAB, and they've created a package that you can download and upload your pictures, and it'll automatically sort through and find matches based on the patterns on the back. It's a little pricey, so we haven't tried it yet. And because we've been catching such low numbers comparatively, it's really easy for us right now to go through and match them. So currently we've only had one match, and it's because it's one of the toads that likes to live in our pollinator garden on the Birkdale property. And it has popped up in the garden and closer down by the river. So he's pretty popular. He shows up often.
That's super cool. Yeah, that's really interesting. So yeah, I look forward to seeing what might come with further development of that technique as well. Okay, moving on to Cindy. Thanks so much for your presentation. It's really good to see over the years what's been developing in your area as well. I'm really encouraged by all the partnerships that you've worked on building, and with governments and other organizations and Indigenous communities as well. I'm wondering if you could just give us an example or a sort of success story or a highlight from one of those partnerships that comes to mind for you.
Yeah, I mean, I think the partnerships are in the early stages still. It takes a long time to really establish good partnerships, but of course the Department of Fisheries and Oceans have been amazing in training and giving us the resources and the technical support that we need for raising our salmon and going out there. And so I think that's probably our strongest partnership right now. One other one that has just developed that I'm really excited about is with I sit on the Upper Bulkley River Roundtable, and from that has come some riparian restoration work where they have hired Rocha to be the communications side of the project, but that also meant that I got to go out and actually help out with the project. And we put in, the crew put in 9,000 willow stems into the Upper Bulkley River in this spring, and we're hoping to do the same in the fall. And that is a partnership between the Maurice Watershed Monitoring Trust, the Office of the Wet'suwet'en, and it's really driven by the First Nations people here, which is really exciting to see the work that they're doing. And they're really the top-notch people in this area who are working on restoration. And so it's been really fun to partner with them and just be a very, very small part in the good work that they're doing. So yeah.
Cool. Thank you, Cindy. Yeah, looking forward to seeing further development in your work as well. Thank you, Christy.
Yeah, thanks so much, Andrew, for drawing out those good questions. And I'm sure it's generated more ideas for questions for in the chat. So yeah, now we're gonna move on to part 2. We have 3 more presentations for you. So first we're gonna hear from Alan. Thanks for putting your video on, Alan. And Alan Constant has been a faithful summer staff biologist with the Rocha team in Ontario for the past few years. And he has a real passion for invertebrates, especially odonates, and which are dragonflies and damselflies. And I've been really impressed at his skill at identifying them. And I'm excited to hear from him and what he's been discovering about our special place in Cedar Haven near Hamilton, Ontario. So go ahead, Alan.
Thanks so much. Yeah, let's get started. So, sweet. Hey everyone, as Christy so nicely introduced me, my name is Alan Constant, and I'm the Conservation and Restoration Technician with Rocha, Ontario this summer. And I'm here to introduce you to Cedar Haven and the wonderful diversity that we have there, with a bit of a focus on the insects because I like those. So Cedar Haven Eco Center is a 96-acre property in Freeholton, Ontario, about 30 minutes north of downtown Hamilton. It's located in the Beverly Swamp, which is a significant wetland. It's part of the Brawny Creek Watershed, the headwaters of that watershed, which flows down into Lake Ontario. So it is a significant site with a lot of invertebrates. And so the 3 groups in particular that we're focusing on are the bumblebees, the odonates, the dragonflies and damselflies, and the Lepidoptera, so moths, butterflies, and skippers. But first off, we have to get into why. Why are we focusing on these groups? And that's because they need us to focus on them. Globally, insect populations have dropped 75% in the last 50 years, 41% in the last decade alone. It's been mentioned in a couple of presentations before, but they're really declining and they really need our help. And so we hope to make Cedar Haven a safe haven. So just quickly getting to the methodologies that we use, it's pretty straightforward, standard things. We do timed area searches, habitat specific. So we pick an ecosystem that we want to target. We go out with nets, we catch some stuff, take pictures, identify, document, and then release. So no killing, no harm, no muss, no fuss. As for sightings, in the past, we've seen quite a high diversity of insects, 30 species of odonates in 2018, 40 in 2020, so good numbers. And some species of conservation concern, like this imperiled arrowhead spiketail in the top left, or the vulnerable double-striped bluet in the bottom left as well. Moving on to bumblebees, we have a high diversity and high abundance on the site. 41 individuals of 6 different species caught within a 2-hour period, which is mind-blowing, including conservation species such as the yellow bumblebee, which is vulnerable, or the yellow banded bumblebee, which is of special concern in Ontario. Moving on to nowadays, this past summer, we got about 30 species of odonates, which is great, including record numbers of the double-striped bluet, which is second from the right there, and some kind of classic Southern Ontario species like the violet dancer, the widow skimmer, and the common whitetail as well. Moving on to bumblebees, we've seen 5 different species, so pretty good, including breeding populations of the yellow bumblebee on the left there, which again is vulnerable. And then some more common species like the brown-belted, the half-black, and the 2-spotted bumblebee as well. So pretty good diversity of bumblebees still. Moving on to Lepidoptera, admittedly the focus this summer was mainly on Odonata and bumblebees, but we still got more than 30 different species, which is great. Some invasive species like the LD moth on the right-hand side, which is not so great, but some really cool ones like monarchs and the snowberry clearwing. Now, one thing that we were able to do this summer is our Bumblebee Workshop, which is one of my personal favorite events that we do at Cedar Haven. where we get community members to come out, learn more about conservation work, insects, restoration practices, and citizen science programs like Bumblebee Watch, where they can upload their observations and help science. Now, even though the summer's kind of drawing to the close, we're still doing some work with monarchs, and we're going to be starting monarch tagging very shortly, partnering with Monarch Watch. So we get these little tags, you see the stickers on the left there, and we catch the monarch, we stick a little sticker onto the wing, and the stickers have codes which we can use to follow the monarchs. Now, for the future, this summer we were looking mainly around the pond due to a couple of the projects that we were doing. And so moving forward, we want to look at some of the other wetland habitats that we have, such as the marshes, the swamps along the stream. We really want to get a nice diversity of habitats we're looking at so we can fully capture the species diversity. And that moves on with the other insect taxa as well. We want to make sure that we're looking at all of the different habitats and we're spending a good amount of time at each habitat to make sure that we're fully capturing the diversity that's present at Cedar Haven, because there's a lot of it and there's a lot to keep track of and a lot to see. And now, this is why we're spending so much time around the pond. We're doing a pond restoration and it's very large scale. We're doing a lot of invasive species removal, planting in native species, water quality testing, vegetation monitoring to make sure everything's changing properly. There's a lot of aspects to this project. And so we want to monitor the biodiversity. Luckily, we can use odonates as indicator species, which is one of the reasons why we do look at them. Their aquatic life stage is very good measure for water quality. And then their adult life stage is a very good indicator for terrestrial habitats, riparian habitat quality and insect abundances and prey species. Moving on to pollinators, because we are doing so many plantings and we're taking out so many plants, putting so many wildflowers and shrubs in, we're really changing the vegetative communities there. And so we want to make sure that we're improving the habitat and we're drawing more pollinators to the site by improving the food sources and the habitat sources there. For the monarch breeding is something that we're hopefully going to do next year. We have a lot of milkweed. And so we're going to, again, be partnering with Monarch Watch and effectively just start raising monarchs at Cedar Haven and hopefully get some boosts to the local population and help monarchs as they go on their journey to Mexico. I just want to take a quick moment to thank all the donors and supporters. We really couldn't do any of the work without your guys' help. Wildlife Preservation Canada, municipal authorities, conservation authorities, specific donors, and just community groups that come out and join us at our events. And that's about it. I mean, thank you all for listening. I really could go on and on and on about insects. So if you have any questions, throw them in the chat. If you want to reach out, feel free to, and I'd love to get connected with you all. Thanks again.
Thanks so much, Alan. That was fabulous. So great to get a taste of what's happening in Cedar Haven. I loved visiting there and seeing with firsthand the diversity, because it's pretty amazing. All right, so now we're going to move along to Graham Peters, who's based in Manitoba. He's our most recent conservation science hire. And he brings a keen enthusiasm getting to know the Boreal Forest Ecology Center property and that ecosystem along the Boggy River. So looking forward to hearing what you have to say, Graham. You can go ahead and take it away.
Thanks, Christy. Here we go. My name is Graham Peters. I'm the Conservation Science Coordinator for Rocha, Manitoba. And today I wanted to share with you some of the monitoring and management projects that we've started at the Borla Ecology Center. I want to talk broadly first about our ecological diversity we see out there, and then focus specifically on our grassland management. So the Borla Ecology Center can be found in southern Manitoba in the Boreal Shield region. This ecozone is classified by its mixed coniferous and broadleaf forests, with discontinuous patches of exposed bedrock. The center contains 220 acres of forest, meadow, and riparian habitat near East Braintree. So to simply call it a mixed forest would be a disservice. And the diversity there is ideal for engagement and learning. As we zoom in closer, we find that we can subdivide our forests into individual forest types called eco-units. In 2006, a report was written that highlighted this ecological diversity. The report describes the center as being made up of 12 unique ecosystem types, with another 5 observed in the surrounding area. So just to give that a little context, I'm going to show you here a map where pink represents meadows, blue shows riparian and wetland habitats, pale green represents aspen forests, dark green represents jack pine forests, gray represents rock outcroppings, orange represents black spruce forests. And over time, these eco-units will shift due to several environmental factors like succession and disturbance. So what we have done is taken updated satellite imagery to approximate how these eco-units have shifted and have since begun ground truthing to confirm the accuracy of these changes. Now, the purpose of this is twofold. First, we can get a better sense of the property and its ecological diversity. And second, this will help us pinpoint places where we should set up more permanent monitoring sites to see how our forests are changing. These sites will allow us to observe and record changes in plant populations and diversity, so that in the future, we can be better informed about best management practices to protect these forest types. We already know that managing forests can support conservation. Fires, for instance, are a natural part of forest cycling. So without them, we see a buildup of dead organic material. In other instances where this has occurred, people have begun simulating fires by removing the dead material. Where effectively managed, this has supported new growth and improved the rate of carbon sequestration. Qualitatively, we know our forests support many plant and animal species. So the next step is going to be affirming our understanding of this biodiversity and how it's changing to determine appropriate responses for conservation. Quantifying this diversity in our forests will be a valuable indicator of ecosystem health. But now that we've looked sort of broadly at the early stages of defining and studying our eco-units, let's focus in on one in particular. Over the past several months, we've been responding to a threat to our meadows. What we've observed is that Scots pine has started to make its way into the meadow. This threatens our grassland's biodiversity and could have impacts on the rest of the ecosystem. Scots pine are especially equipped to survive in high sun exposure, so this leaves our meadows especially vulnerable. Our objective then was to remove these invasive trees, but because our workforce is primarily made up of volunteers, it's important first to mark them in order to avoid any accidental targeting of native species like tamarack or white pine. Although this marking required a lot of work, it has also provided an opportunity for data collection. We collected information about the size of the Scots pine, measuring the trunk circumference at 1 meter, and as well as the status of their cones, shown here as flowers, immature cones, and mature cones. What we found was that the larger trees more frequently have cones and flowers, making them a larger reproductive threat moving forward. However, we also found in the meadow that we have far fewer large reproductive trees and a lot more of the immature trees that aren't reproducing yet. So we've begun the process of removing the Scots pine. It's critical to remove the cones so they can't reproduce. The rest of the wood we're able to repurpose because it poses no invasive threat. Our grasslands are especially susceptible to invasive species like the aforementioned Scots pine, as well as vetch or thistle, or Canada thistle to be more specific. However, they also provide important habitat for a variety of birds and insects. This has spawned the need to better understand how to invest in the meadow and why we should do so. Now, some might express concern at the idea of culling trees when we're encouraged to plant trees in order to mitigate climate change. However, effective grassland management can also support carbon sequestration, and an effectively managed prairie is valuable for biodiversity and can better adapt in a changing climate. We know our meadows house a great many flowers and insects, which support a variety of animals. By actively combating invasive species and supporting native species, we hope to be able to preserve this diversity. We know the Boreal Ecology Center is full of diverse ecosystems, so our focus is on setting up projects to effectively monitor and manage them. This will help us better promote conservation practices in the future. And I just want to give a big thanks to our various private donors, as well as the Drew Foundation and Project Learning Tree for their support. So thank you.
Thanks, Graham. That was fabulous. If we, one of the drawbacks of not being in person, of course, is the big rounds of applause that we're not able to hear, which you would be hearing right now. So I have to use your imagination. But thanks so much for sharing. I am gonna ask you to just take your video off and then shift over to my presentation. So I'm Christy Jouteau. I had the pleasure of serving on the conservation science team for the past 10 years. And I'm so grateful to work alongside such exceptional people that have already been sharing with you, and also with the amazing support of all of you fine folks who are listening today, and all the support of our volunteers and donors. I'm just really humbled to be able to participate in this good work. So I'm just going to share my screen with you here. So thank you for sticking with us. We've highlighted projects tonight from vertebrates to invertebrates to plants, and now to a project that's very close to my heart, back to the Pacific coast, looking at some of the smallest living things, bacteria. So this project touches on the overlapping triple challenge we're facing at this present moment: biodiversity loss, climate change, and food security. The UN are hosting 3 summits this fall on these topics: food systems in September, Convention on Biological Diversity in October, and climate COP26 in November.
Hey, Christy.
Yeah.
Sorry, I'm interrupting because I don't see your screen.
Oh.
Somehow the sharing isn't happening just yet.
Thank you for interrupting me. Should I? Here, okay.
Oh, there, something's coming now.
Okay, that's
there. I see Julia.
Oh, good. There's Julia. How's this?
There we go. That's SDGs.
Great. Okay, I'm just gonna, so this was the previous picture. Here, give you a little what you missed. My title slide. This is the triple challenge. All right. So many agree that we are in a critical point in history with an opportunity to pause, reflect, and shift the way that we live on this planet in ways that renew and establish good relationships between people, the rest of creation, and with our creator. We know that these crises that we're facing are felt most by the most vulnerable populations. So we've been talking about sensitive species like Western toad, Pacific salmon, the yellow bumblebee, and this also relates to people. This is my friend, Chief Harley Chappell, who leads the Semiahmoo people of Semiahmoo First Nation. And Semiahmoo means lowering to feed. And that refers to the way that they've always received their sustenance from the long, shallow tidal flats of Boundary Bay. They have a traditional saying that they share with many other coastal First Nations: when the tide is out, the table is set. Unfortunately, shellfish harvest is prohibited in Boundary Bay due to contaminated water. In fact, most of the Pacific coast is closed to harvest, as shown in red on this map. And shellfish are managed federally by DFO, and their mandate is to protect human health, but they don't currently have a mandate to restore shellfish harvest in areas where it's closed. So we're talking about Boundary Bay, a key biodiversity area, hosting millions of shorebirds, rich eelgrass beds, and used to produce over 50% of BC's commercial oyster production. And we're focusing our attention on the Tataloo, or the Little Campbell River watershed, because it has been found to be the greatest contributor of contamination to the bay. And so this is a map highlighting the many different jurisdictions in the watershed, 4 municipalities across 2 countries, and Semiahmoo First Nations right at the estuary. So this water is contaminated, but with what, you might ask? So many things, but what we're focusing on today is Fecal Coliform, a type of bacteria that's found in the gut of warm-blooded animals, used as an indicator of pathogens or disease-causing bacteria. So where is this bacterial pollution coming from? Many different sources, including pet waste, failing septic systems, sanitary pump stations and sewers, livestock manure runoff, stormwater, and each of these sources of contamination have different organizations that are responsible for managing them. So BC Ministry of Agriculture is responsible for manure management, and Fraser Health Authority for septics, Metro Vancouver for sanitary, pump stations and systems, municipalities for stormwater, and the general public for pet waste. So this is a complex problem. In order to address it, Rocha has helped to revitalize a transboundary working group called Shared Waters Alliance. And we're standing alongside Semiahmoo First Nation to gather these many stakeholders together, acknowledging that we need each other to accomplish this vision of a healthy Boundary Bay. One of the key actions coming out of this Shared Waters group is monitoring the water quality to better understand where the contamination is coming from. So here's a map of our 7 sites that we've been monitoring since September 2020. Funded by City of Surrey, Sitka Foundation, and BC's Healthy Watershed Initiative. Here's the same map showing our sampling results since September. These are mean fecal coliform concentrations. So the high coliform concentrations are larger and redder, and the lower concentrations are shown as smaller orange or yellow circles. You can note that there are no green circles on the map, meaning that none of the sites are currently meeting the shellfish guideline of 14 CFU per 100 mils. Most sites are meeting the recreational guidelines, so it's safe to swim, except for Fergus Creek, the big red dot, it's a major tributary to the Little Campbell Tataloo River, and it is the most contaminated so far. So what are we doing about this? One thing we're planning for the fall is to increase the number of sites and frequency of sampling in Fergus Creek to better understand the sources of contamination there. We're working with partners in Langley to increase sample sites upstream. And we're continuing the Shared Waters Roundtable meetings to engage the different organizations to address various contamination sources in their jurisdictions, and restoring habitat along the river and its tributaries to improve biodiversity and also filter runoff before it reaches the stream in the ocean. So is success actually possible? Could we clean up the bay so that to a level that would actually allow for shellfish harvest? We do have good news stories in Drayton Harbor, and with the Lummi Nation and the Tsleil-Waututh Nation are both able to harvest shellfish in their traditional territories. So we have hope that it's possible for us too, for Semiahmoo. Chief Harley and I have invited all of the municipal elected officials from municipalities around the bay to a gathering that's happening in September to talk about our shared concerns and interests in clean water and Indigenous reconciliation. So those are the next steps for us in this project. Thank you so much for listening and for partnering with us on this important work. Thank you. There we go. I get the prize for the most technical glitches. So thank you. Okay, I'm going to now invite Rachel Krause to join us to ask a few questions of the last three presenters. So if the Alan and Graham could highlight or put on their video. And there's Rachel. Rachel's one of our science advisory team members, and she's based in Manitoba and has been as a prof at CMU and the Canadian Mennonite University. Is that right? Yeah, so thank you, Rachel, for being willing to ask a few questions of us.
Yeah, thanks for this invitation, Christy. It's always fun to see what kinds of conservation projects have been happening across the country, and I am missing Sean's dessert this year. So you can
Yes,
let him know
as
that.
we all are.
But no, really terrific presentation. So I'll go in the order that you presented, the three of you. So Alan, you're the only one here that I don't personally know, so it's good to meet you online here and to hear about some of your work at Cedar Haven. When I think of Hamilton, I don't necessarily think of great biodiversity, so it was great to learn a bit about what all you're finding there. And it sounds like you've done an enormous amount of really dedicated work there. So I just want to congratulate you on that. I have lots of questions, but I'll just ask you one, and maybe others will pick up some of the other ones that I've been thinking about. This pond restoration that you were talking about, sounds like that's pretty major work. So I like the idea of using odonates as an indicator species there. I'm wondering if you've observed downsides to the disturbance in the meantime as you're doing this restoration work, or if there are ways that your team has tried to mitigate some of that, or is that not a concern at all? And when I think of restoration, I think of pulling stuff off and putting stuff in and disturbing sediments and water.
Yeah,
Yeah.
it's definitely a bit of a balance. It can be a little hard at times, but you kind of just need to keep telling yourself, we're doing a little bit of harm right now to do more good later on. So far for the restoration, a lot of the sort of invasive species removal in particular has been based on the aquatic plants. There is an aquatic plants, curly leaf pondweed, that's quite pervasive in the pond. And so we've been able to get that out with a couple of special tools. It's essentially just a big metal rake and razor that we just toss in and pull the weed out. And thankfully it hasn't had too big of an impact on diversity or on abundances of anything. There has been a slight drop between 2020 and 2021. Last year we saw about 40 different species of odonates. This year we saw low 30s or so. So there is a slight decline, but it's I can't exactly contribute that totally to the restoration project. There could be other factors that I'm not fully aware of. And in general, we're just trying to preserve little aspects of the habitat that the odonates can use. If we're noticing them using certain stretches of the pond for breeding habitat, we try and avoid those ones for the time being and instead focus on other sections of the pond. And then we're kind of hoping to do this restoration process over a number of years so that it's not just one really abrupt transition that could devastate their breeding habitats.
Yeah, it really highlights the value of long-term monitoring, doesn't it?
Yeah,
More than 1 year, which
absolutely.
which is real value of your work there. Yeah, thanks for that. Really interesting. Graham, good to see you on the screen here. So I'll just full disclosure for everyone, I do know the Boreal Ecology Center a little bit, and I worked there with Graham actually when he was a student with me. So it's nice to see how the work there has progressed. You talked a lot about meadow work that you've been doing and work looking at this invasive Scots pine that I hadn't been aware of beforehand. So I was really interested to learn about that. So thanks for that. I'm wondering if, I think with the Boreal Ecology Center, it's a little bit newer in the Aurashakenda family, and the conservation science work there is a little bit earlier along. I'm wondering if you could share with us maybe one conservation project that you would dream of or imagine for the center in the future.
Just one? Oh my goodness. That might. Well, I guess while I've been highlighting the meadow, a lot of what I've been thinking about when working out there is doing some invertebrate sort of sampling, because what we see a lot of, just qualitatively, there's a lot of insects flying around in the meadows, and our meadow, if you take a look at the map there, it is really close to our river, our riparian area. So we have different life stages for these insects that are happening really close together, which lends itself to the idea that there should be a lot of insect diversity there. So really, it would be, I think, of great value to actually concretely learn about that and do some sampling, and just just really get to know the insect diversity out there. That's sort of one of the projects that's been on my mind recently. Every time I go out there, there's another 2 or 3 things. I think Scott can attest. Every week I come back and say, hey, I thought of this thing that we should do out there. But yeah, that's probably the most recent thing that I've been thinking about with the meadow work.
So it sounds like maybe Ellen needs to make a trip out there next summer. Help you set something up. You know, that's really interesting.
Be happy to have it.
Christy, good to hear about the bacteria work in the Little Campbell River. So many years ago when I was an intern, I worked
a lifetime ago, yeah.
A lifetime ago, and I remember doing some of this monitoring work. So on one hand, it's good to see that is still a focus. On the other hand, these are slow changes that we're looking to make, aren't they? Which is can be encouraging.
Perseverance.
I'm wondering what do you think it's going to take to restore Boundary Bay to a state where the Semiahmoo people can go back to their traditional shellfish harvesting? Probably a number of things, but what do you think are the top few?
I think, I've been thinking about this a lot. But I think one of the critical pieces is actually, I guess, establishing or acknowledging First Nations rights and seeing reconciliation actually move forward. So recognizing that the health of the river and the bay, it's actually their right to traditional harvest, because right now there's a real gap in mandate or legislation to improve water quality to shellfish harvest level, right? The federal government doesn't have a mandate to restore, they just are protecting, and the provincial government is responsible for the fresh water, but they're not, shellfish isn't part of their mandates. They're not looking to that stringent of a standard, and neither are municipalities or the regional district. They all have different kind of focuses, but if UNDRIP or the the provincial legislation related to the United Nations Declaration of Reconciliation for Indigenous People is actually implemented, where they can use that legislation to actually hold the various jurisdictions to account to that higher level. Then I think that's a critical piece, and that's going to be a long game as well. Like, this is a long game where we are we're in it for the long haul. But I think that's a critical piece, which is part of the reason why we're engaging municipalities right now, elected officials around that location with First Nations leaders to try and highlight that piece of it. Like, this is it's all of our shared responsibility to improve water quality for First Nations, and that really, that's a win for everyone, right? And so yeah, not an easy solution for sure, but
yeah,
we'll work towards it.
Highlights again the importance of longevity in conservation work, and it also makes me wonder if we thought about reconciliation with Indigenous partners on the land and all of our sites at Rocha, Canada. What would that look like, and how would that shape the work that we all try to do together? Anyway, thank you. This has been really really wonderful.
Thanks, Rachel, very much, and all of you for participating so far. I think it's time to invite questions from the chat. So Rick's back on, and I guess everybody can, all the presenters can put their video back on. We can all get here on the screen, and Rick's going to facilitate some questions from the chat.
Okay, well, there's been some great questions from the chat, probably more than we're going to be able to get to, but I'll just pick a couple. And if your question doesn't get discussed, forgive me. Nothing personal, just time. So I had one about I've wondered this myself about I'm going back to the toads. So Laura, you might be the one to ask about this one. Can the fencing be made permanent, essentially? Like, what would it take to, it's a ton of effort to put it up just for the migratory season. Like, what would it take to make something that is more permanent and presumably then more effective? What are the challenges for that? Like, again, I'm not the conservation science guy, so like, I don't know.
Yeah, it is a challenge to build. Our poor interns had quite the work cut out for them early this summer. We have started to use like a corrugated plastic piping that we cut in half. So you have like the half moon shape, and then we kind of bury that edge in, and it's a little bit more permanent. Right now, the fence is built on Olivia's property. She's lovely. She's very concerned about the toads, and we're hoping maybe she'll let us leave part of it up throughout the year. But the issue is it dips down quite a ways into the wetland, so it seasonally floods. So a good majority of the fence would be underwater. So that's definitely a constraint. But there are different things if we were able to work more with the city and city planners about looking at like more underground crossings for the toads, and building more permanent fences along roadways to help direct them underneath the road. So that's kind of long-term future hopes and dreams of more city planning and involvement from the township of Langley. So yeah, dreams.
I'm sensing a theme here. The long-term thing is definitely a theme. Although a fascinating one that you're drawing out too is the interaction with landowners. Like, this is inherently partnerships are an inherent part of this work. They're just crucial.
Yeah,
so
for sure.
Alan, there's one question that came in from the chat here about pollinators. Have you noticed how much some of the invasive plants are used as pollinators. So if the invasive plants are lost, that's, you know, a common restoration tactic. How much of the pollinator is going to get affected?
So thankfully, in short, not many. A lot of the invasive plants, like I said before, that we're removing are aquatic. They're fully submerged, and so they're not going to be used for pollen. Because we are in Southern Ontario, we do have kind of the classic exotic species. You know, we have like the different clovers. We have bird's foot trefoil, Canada thistle, the sort of big invasive flowering plants. But there's really not that many around the pond that would be affected by the restoration process. And ultimately, we're actually adding more flowering plant species than we are going to be taking out. So it should be okay.
Okay. Well, that sounds sensible. Okay, I got one here from Michelle. This is an interesting one. It is asking, how do those swallows predate the wetland invertebrates? Now, I immediately think, okay, they're flying around. I see them flying over the river all the time. So they're catching them in the air. But then the question asks something I hadn't thought of. Well, do the swallows also eat the larvae of various types? Do we know anything about how that works?
As far as I know, they're just eating the adults as they're flying around.
Okay.
So, yeah. The larvae are important, and they're the most, I mean, they're going to be affected by things like water quality and temperature. So they're an important life stage for determining the abundance of the adults as they're flying around for the swallows, but the swallows are directly eating the adults.
Okay.
Good question, though.
All right. Cindy, I'm planning to come visit you next week. I don't think you said anything about this, but I'm prompting you. You got to tell us what's happening next week.
So we're having our grand opening for our nature center next week. Yay. It's been about 7 years in the making, and we are just finalizing the last bits of paint and rocks and landscaping tomorrow to be ready for Wednesday. But yeah, I'm looking very much forward to not fundraising and not constructing, so that I can get to the business of what I actually want to do, which is education and conservation. So it's been a long time, but I really feel like we have a space now and a place that we can do this work really effectively, and it's going to be super fun. I'm really looking forward to you guys coming.
Yes, I'm looking forward to being there. Graham, so Rocha is famous, at least within the Rocha global family. Like, it started with Peter Miranda Harris, and Peter's this like totally manic birder. Started with birds. It was noteworthy when you were asked for your dreams for the Boreal Ecology Center, you didn't include anything about birds. Like, I'm going to give you another chance. Come on. Anything to do with birds happening out there?
So I'm not to argue with you, Rick, but I would say that if you follow the insects through the food chain, that you would get to birds. So I'm glad you asked me. In terms of research, we've done a lot of early thinking about again, getting that good quantitative data. Right now, we have seen a lot of different birds, and we've planned some different sort of birding events and sort of ways to invite people who are more knowledgeable about birding than I am to come down and really explore the area. One of the reasons I was bringing up this insect thing was because we have seen a lot of insectivore birds out there, like Kingbird was a recent one that we've seen, and has led to this thinking about protecting the prairie, as well as I think I showed in my slideshow a nest that was spotted that we believe belonged to clay-colored sparrow, another meadow nesting bird. So yeah, it's sort of snowballing as we learn more and explore more.
Cool. Here's a question that might apply to a number of you. And I'm drawing on a few different questions from the chat that had a bit of a theme here. I recently participated in a BioBlitz, and for the first time, I was getting to use iNaturalist. It's one form of a kind of it's an app. It's a kind of collective database that many people can contribute to. Some of the questions asked such things along those lines. Hey, are we connecting into the database for the I'm going to get the name wrong, the odonates? Oh, come on, help me out. Or other, like a pond watch program, or yeah, okay, I'm sure there's others. Like how are we, or how are your projects connecting in with these kinds of larger databases, so it's something that we can contribute to beyond Rocha's work itself? This is directed to whoever wants to reply, so come on, not all at once.
I can start things off, mainly because the odonate. So for all of the insect observations that we make, when we're photographing them, we are uploading the identifications to iNaturalist, just to help further the overall kind of understanding of the biodiversity on the site. And then, like I mentioned in the slides as well, we are working with Bumblebee Watch, which is another citizen science program. And so we're uploading our Bumblebee data there, just because it's so much more easily accessible to entomologists and decision makers and things like that. So we are connecting with citizen science applications at Cedar Haven.
Yeah, I think the Ontario team and Cindy as well really led the way with using iNaturalist. And here in Brookstall, we've just kind of gotten started using it. But it is a really great tool to engage anybody to get to know their place, because you can just take a picture and it prompts you with ideas of what that could be. And it's a really great way to document the diversity of an area. So we did use it just recently. Michelle helped to coordinate a BioBlitz in the forested areas around Brookstall, and these forests that are at risk of being turned into industrial land, at least parts of them. And so we, hopefully it can be used as a tool to demonstrate the biodiversity values of those forested areas. And can see lots of other applications.
I'll just jump in and say too that we do weekly bird walks in the Little Campbell Watershed, including Brooksdale. That's one of our sites. And these bird walks have been going back since, since Rocha Canada started. So is it 2001 maybe? And those data all get uploaded to eBird, which is a global online platform for uploading bird lists, and there's millions of observations on there. So we have 5 sites that we rotate among for the weekly bird walks, and all of that bird data goes into eBird, including all the birds seen at Brooksdale. So that's one way that we're contributing to citizen science, as well as using iNaturalist more and more with our interns. And we've experimented with doing some competitions among the interns to try to get them to upload more sightings to iNaturalist, which helps them to get to know their place and also helps us to catalog just what's there at our Brooksdale Center.
Thank you all. I've got one more question I couldn't resist. I know we're nearly done, so this is going to have to be the end. But I was fascinated by this question that was for Christy. The Fergus Creek big red dot. What's going on there, and how are we going to find out what's happening? You mentioned it a little bit, but the question actually said, "Have you dug into DNA-based source tracking and the verification of the fecal coliform results?" Any other, I'm not sure if you want to name names, but you got any ideas about how we might get to sleuth to the bottom of that issue?
Yeah, I think that was, you put two questions together there. And yes, Fergus Creek, we're going to need to do a bunch more sampling to pinpoint where the sources are coming from. In Whatcom County, they've had great success in just wherever there's a hot spot to just increase the frequency and the number of sites in a particular area to be able to pinpoint the source. And they found that's been actually even more effective than using DNA analysis or bacterial source tracking, which they've also tried. The bacterial source tracking is more an expensive procedure, and it tells you perhaps what kind of creature is that bacteria is coming from, but it also it doesn't give you the relative quantities. It's not quantitative. It just gives you the number of potential sources, which can be informative, but it's because of the cost benefit. It's actually in Whatcom they've found it's more effective to do the cheap fecal coliform concentration, grab sample data frequently, and you're able to really get to the source. So I'm not sure where it's coming from. Could be the golf course, although no, actually, because the site is actually upstream of both the golf courses. So it's not the golf course, Pamela, but I think it's coming from upstream, and we have found that stormwater is quite contaminated, and I think that's going to be a critical piece of just to add to maybe my answer from earlier of what needs to change. Stormwater needs to be better managed and treated because it's really contaminated. So yeah, we can't assume it's clean.
Well, that's a cautionary tale, because like just where I think I know the place you're talking about, there's a whole new subdivision put in there just in the last right now. So stormwater makes a lot of sense. None of that, like it's not landscaped well, like anyway, it'd be lots of runoff there, I'm sure.
Yep.
Well, I'm going to have to call it there. Thank you all for your participation, and it's so fun. And I mean, in conservation work, some of the trend lines are discouraging. We saw a few of those, but it's so fun to hear all the work that's being done because this is a part of trying to address those trend lines, trying to make it better. So all of you are on the front lines of the fieldwork of that day in and day out, and I'm just so grateful for your taking the time to share some of that with those of us that aren't in that role. Thank you all for those that joined us tonight. It's been great to have you. It's been fun to experiment with this digital online version of our science presentations. I'm sure we'll evaluate and decide how we might want to do that in the future. If you've got opinions about that, you probably already have received a note in your inbox inviting you to participate in the evaluation form for this Rocha talk. So that would be a helpful place to register your feedback and your input on that. If you're interested in other Rocha talks, you can go to Rocha's Canada's YouTube channel and you'll find Rocha talks, but you'll find all sorts of other video content on there. Everything from like a whole series on birding that actually came out of the Cedar Haven folks last year, for one example, but lots of other things too. So check that out, rocha.ca. You'll find us on Facebook, but the best thing, especially since COVID is starting to well, I want to say wane, but at least a few more restrictions are being lifted. Come and visit. So as it's safe and appropriate and you are able, please come and visit. That's always the best thing. This has been recorded and there will be a recording posted at some point. So you can look for that and pass it on to others if that's of interest. But stay tuned through our e-news and you can get signed up with that if you want to find out about future talks. There will be more coming in the fall. I don't have anything specific right now to share, but there definitely will be some. So we look forward to having you join us again sometime. And with that, I think I'll say goodbye. Thanks, everyone.